Okay, morning guys. Can you let me know if you can hear me okay? I've got the different mic on and YouTube is saying the audio stream's not brilliant, so I just want to make sure that you can actually hear me okay. If I don't hear anything, I will carry on as if you can hear me without a problem on this one. No, let's just carry on then. Okay, so I thought today... brilliant, thanks Poppy. So I thought today we'll start off with cells. This should be some revision. I've sent a link out to year 9, I've sent a link out to year 10, so it does cover most people. Cool, morning Tanishka, Katie, Mia, good to see you up. I thought I'd do a slightly later one, although it's still technically 10 o'clock as previous, but obviously with the clocks changing now. I thought I'd do 11 o'clock, and we'll sort of probably stick to 11 o'clock from now on, if that's okay with everybody else. So, working our way through then, we have two main types of cells that we need to be looking at. We have our prokaryotes, hi Katie, and we have our eukaryotes. So, let's just bring this into pen mode. There we go. Okay, so we've got two types of cells. The one that we're most familiar with is going to be the eukaryotes. So this is any multicellular organism. We're looking at animals, we're looking at plants, we're looking at yeast. But we're looking at multicellular. Morning, Ollie. When we're looking at prokaryotes, we're looking at bacteria. And there are some sort of structural differences that we'll see as we sort of work our way through with this one. So we need to make sure we know what those two words mean. You don't need to go into loads of detail, but this is multicellular. And this is generally single cells. Okay. Now, hang on. Let's move that up. Still not working. There we go. Move it up out of the way. Brilliant. So, structure of the animal cell. Let's keep it nice and simple. Let's do what we've been doing before. I thought I'd bring this diagram in because it's actually goes into a little bit more detail. When it shows up, there we go. Goes into a little bit more detail. It's the sort of thing that we might start seeing, especially if we can start going into A level. We don't need to know about a lot of the other structures at GCSE. But it is a sort of diagram you will need to know in more detail as we go through. So we should be fairly familiar. We've got our cell membrane. Remember, the function of these is as important than the actual name itself. So we need to make sure we can talk about what the structure of these are. So cell membrane keeps everything in the cell and, more importantly, controls what's going into and out of that cell. We should have our cytoplasm. We've got a nucleus. This one's already been identified, or it's been diagrammed here. We've got our mitochondria. And these little dots here are the ribosomes. So the cytoplasm is the sort of jelly-like substance inside the cell. It's where most of the chemical reactions happen. And it's really quite important that we get the concentration of that right. If the concentration is wrong, the chemical reactions don't work properly and we start feeling quite lousy, or the organism starts not responding properly. So when we start talking about dehydration, in a lot of cases it's about trying to make sure that the cytoplasm inside the cells is actually the right concentration. So the nucleus, we know, hopefully know, contains the DNA. So the DNA is the genetic information, deoxyribonucleic acid, and it's the information that tells the body how to make all of the different proteins that make us up. The mitochondria, most importantly, because this comes up a lot, is the site of respiration. And we should know that, and we should know the formula for that. I'm not going to talk a lot about respiration now because it's the topic that comes up later on. But it's one of those sort of key topics that is really, really vital you get your head around. And the ribosomes are literally responsible for putting together amino acids to make proteins based on the information in the DNA. So they're only tiny, but they're really, really important. If we look at bringing in a plant cell now, I think I've got a plant cell on here. Yeah, there we go. So all of these diagrams, or most of these diagrams, have been taken straight from the exam board, so straight from AQA. So I'm trying to use those as much as I can. One, we're allowed to use them, and two, it gives you a bit more sort of familiarity with what's going on. Good morning, Olivia. So cell membrane should be the same. It's this inner one here. Cytoplasm into the cell there. I'm just ignoring the arrows for the moment. Nucleus. So a lot of these are the same. We should still have mitochondria. In this case, I think they've probably put the mitochondria down as these ones. Maybe. It's difficult to see. And we will still have the ribosomes will be probably dotted around inside. So we can say we've got the ribosomes as well. Oh. Just zoom back out. There we go. So it's quite important with these that we also know the differences. And the main differences, and let's do them in a different colour just so we can see. We've got the cell wall. Cell wall is made of cellulose and it gives the plants structure. So plants don't have a skeleton like we do, which means if it was just made of cells, they would just sort of pretty much be a blob on the floor. They'd contract and expand, but they wouldn't be able to get the heights. They wouldn't have the structure that they have now. So the cellulose gives it the structure, and it also is quite important when we get onto osmosis. And we'll get to that in a minute. We get to osmosis a bit later on today. We've also got chloroplasts. So chloroplasts contain a green pigment called chlorophyll. Now, this is the opposite of respiration. This is our photosynthesis. From a GCSE point of view, it's exactly the same reaction, but going backwards. So if we know one, we know the other ones. And this is our permanent vacuole. And the vacuole is where the cell sap is stored in the plant. Just completely aside from this, it's worth just making a note, just because it's one of those things that comes up sometimes and people sort of don't really get their head around. When we're looking at oxygen transportation, one of the things that we need in humans is iron. So we know that iron is the pigment in red blood cells that transports oxygen around. When we're actually looking at plants with photosynthesis, they've got a different mineral, which is magnesium. And the main pigment, the main mineral in chlorophyll, which helps it do its job, is magnesium. So a lot of plants, if they have magnesium deficiencies, they're not as green, they look a bit yellow, and the photosynthesis doesn't happen properly. So it's just one of those that comes up every now and again. Okay, just shift that up a little bit. So, another cell then, as we're talking about, those are the two main ones for eukaryotes. If we're looking at prokaryotes, I can drop in a picture of a bacterial cell. So, some key differences here. The bacterial cells are too small for mitochondria, so they don't have mitochondria. They do have, let's use a different colour this time, we do still have a cell wall, very similar to plants. I could just write that on A. We've also got a cell membrane. They're still going to need to control what's going in and out. But there's no nucleus. What we have instead are plasmids, which are small circles of genetic information, and we have this loop of genetic information here. Rather than having the sort of pairs of chromosomes that make us up, we still have cytoplasm. And if I just extend this one slightly here, we've still got ribosomes in there, because it's really important that actually our bacterial cells can still make proteins, because actually a lot of the enzyme, in fact all enzymes are a type of protein. A lot of the other chemicals that are made are protein based, so we're still going to have ribosomes in there. So some key differences. Sometimes what you'll see on here is you will see what they call a flagellum, which is like a tail. Oh, sorry, hang on. We'll see a flagellum, but not on all of them. But yeah, it's worth having that down as well. So if we're looking at the key differences, key difference from an animal cell through to a bacteria, much, much smaller, too small even for mitochondria. They have a cell wall that animal cells don't have. They don't have a nucleus, but they do have genetic information, but it's in a loop and in plasmids rather than in a sort of in a nucleus. When we're looking at difference between bacteria and plants, similarity with the cell wall, but difference is we're not going to have chloroplasts in this case. Again, they're going to be too small. There's no mitochondria, there's no vacuole, there's no nucleus, but they do have the similarity of the cell wall. So hopefully that makes sense. Flagella, tenacious generally is just for movement. Now, it's similar when we're looking at sort of sperm cells. They don't say, right, I'm going over there, but it does mean that actually by moving, they can actually just move themselves in random directions, which helps them get to other places. So again, when we're looking at sperm cells will have the tail on the cell that is moving randomly, but they're not suddenly going, there's an egg, I'm going to go to it. They're just going to move in random directions, and hopefully they're going to hit the egg, which is why so many of them are released. But it's for movement. Good question, though. So if we're looking at specialised cells, let's look at a few of these. So we know that the cells we've just looked at, but let me just bring that back in again. The cells we've just looked at, these would be types of stem cells. And I'm going to talk about stem cells a bit more, probably in a different session, just because I can go over and do a recap on this one, because we're probably only going to cover about half of cells today, depending on how far we get, because I'm going to cover some triple content a little bit later on as well. But we'll see from that point of view. But these would all be stem cells, so they're undifferentiated. And if you think about all of the different parts of your body, the different tissue types, they're all going to be made of different cells. So when we look at our sort of basic animal cell and our basic plant cell, they're not going to be able to do all of the different functions of the different tissue types. So these cells have to become specialised. They have to differentiate for their job. So at the moment, and I'll just put this up here as a note rather than the fact I'm actually going to talk about it too much. But these are stem cells. Okay, which means they are undifferentiated. Now, in order for them to do their job, what we have to do is we have to make them differentiated. And you need to be able to talk about some of the specialised cells. You need to be able to talk about what makes them special, as in how are they different from those stem cells we've just been looking at. So let me just drag in this one for a minute. So, just quickly, and I'll bring a few more in a minute as well. We know we've got our sperm cell. If we look at the sort of main differences, we're generally streamlined. And remember, we don't just want to be able to identify, we need to be able to talk about how it's an advantage. So the more streamlined we are, the faster we're going to be able to swim. The easier we're going to be able to swim, so less drag. We have enzymes. So the enzymes in the head of the sperm, they're going to help it break through the wall of the egg to hopefully fertilise it. The nucleus. The main difference is not the fact it's got a nucleus, it's the fact that it's actually only got half the genetic information. So it's got half the amount of DNA, half the number of chromosomes, because it's going to get the other half when it combines with the egg. So it only needs half now. we have mitochondria here. And the tail, very similar to the flagellum in the bacteria, is already identified. And the mitochondria are going to be providing energy for that tail to propel the sperm to help it swim. So just in one simple cell, we've got quite a few differences. But actually, we've still got a cell membrane. We're still going to have cytoplasm in there. The rest of it's going to be very, very similar. So this looks like epithelial, to be fair. Difficult to tell with this case. We know it's cytoplasm. We can't see many structures. They're not labelling much in the way of mitochondria. Again, this was straight from an exam question. So I haven't looked at the exam question. I'm just taking out information as I would see it. And hopefully as you would see it if you had to sit the exam on this. It could have lots of ribosomes if it was in a gland, but they haven't labelled that on there. So in this case, we're probably looking at some form of epithelial cell, which is like a skin. Yeah, we can't really tell much more about this one. The root hair cell, though, we're obviously looking in plants. And the key thing here is this protrusion is increasing our surface area. Now, I am going to talk about diffusion later on in this session. And increasing the surface area is going to increase the diffusion. It's going to increase the rate at which we can transfer substances into the root hair. So that's the main one on that one. Notice, and this is quite important for a root hair cell, there are no chloroplasts. Okay? It doesn't need it. It's underground. And I've seen students write that before, make mistakes. I've also seen a certain dietary, I'm not going to credit her with calling her a scientist, but a certain dietary TV celebrity that has sort of talked about the fact we eat greens because it can help oxygenate our blood, even though there's no light in our blood sort of system, in our digestive system, so it sort of just doesn't work. And it's the same principle here. There's no point in having chloroplasts in root cells because they're underground. They're not going to get any light. So we could say an adaptation is the fact they just don't have chloroplasts. Finally, we've got a diagram here that looks very similar to our sort of stem cell up there. And realistically, it is. And this would be from a palisade cell. So I'm not going to do the structure of the leaf too much at the moment. I'm just going to drop one in just so you can see. And I'm going to come back and do this when we do biology 2. So if I just move this down here for a minute. So these palisade cells are here. And if we were going to say about an adaptation, the main adaptation is they're actually, because of the way they're lined up, they've got a fairly large surface area, they're loaded with chloroplasts. And the idea is that the bulk of the photosynthesis in the plant is going to happen in this palisade layer. So the key thing is when we're looking at our specialised cells is that we're trying to identify what's different so that we can then go through and we can actually start talking about, okay, how is that different going to help it do its job? And that's the sort of key thing in any exam question. What's different? Why is that difference important? And we'll come back and we'll look at some specialised cells when we do biology 2. Biology 2 goes into organ systems, it goes into tissue types. And we can really start looking at the function of the organ and the tissue then and start looking at why those cells are a bit more important. Okay, transporting cells then. So let me just have a quick look on that one. Yeah, so one of the exam questions that came up, I can't remember which year ago, it wasn't that long ago, but it was either done really, really well or pretty much not at all by students, was this one. Let's make it a little bit bigger, that again, too blurred. So if we knew a bit about diffusion, the question was generally done really, really well. If you could try and, I suppose if what I'm saying, if you knew a bit about it, if you knew what diffusion was, you could probably work out enough to get at least 4 marks on it. So people either did at least 4 marks or they got pretty much nothing at all. So let me go through some of the background around this one. I'm not going to answer this one because this is going to be one of the questions that I'm going to drop into Google Classroom later on for you to have a go at by yourself. So I'll give you the tools for answering it now, and I'm going to drop it into Google Classroom in a way that you can actually answer it, and then I'll have a look at some of the answers that you've done. So that'll come on pretty much straight after this lesson. Right, so we're going to have a look at diffusion, osmosis, and active transport, because they're pretty much related. But getting your head around diffusion is absolutely key here. So, diffusion. We're looking at the movement of substances from an area of high concentration. I should just type this, it'd be much quicker than me writing it. To an area of low concentration. Okay, so in terms of the key sort of bits on this one, we've got this idea then, we've got our concentration types, and I'm going to come back to that one next. Low concentration, and by substances, we're looking at fluids. So we're looking at pretty much any substance, whether it's a gas or whether it's a liquid. And there's in living things as well as, I suppose, outside. I mean, diffusion comes up in physics and chemistry as well. So it's a huge topic. And to get your head around diffusion properly, we must have a bit of an idea of kinetic theory and chemistry. So it's one of those sort of connected topics. And this is the idea that as we give particles energy, they start to move faster. If we make particles move faster, they gain energy and they get hotter. And how they interact when they're moving faster is really quite important for diffusion. So if you think about this idea that if we had lots of sort of particles, let's just do it as a gas for a minute. Let's just say we sort of sprayed some form of aerosol around here, and they're sort of moving around and they're bouncing around. If they're crowded together, we're going to have a much higher frequency of collisions. So we've got a high frequency of collisions here. Now, every time they collide, they're going to bounce away from whatever they've collided with. So gradually what they're going to do is they're actually going to spread themselves out. How many have I done? 1, 2, 3, 4, 5, 6, 7. Okay, they're still going to be moving around. But actually, they're going to be moving around until we have the lowest number of collisions possible. And you can sort of think about this in a lot of cases. I often do this one when I'm talking about young kids. You can imagine if you had a load of young kids, you had them sort of blindfolded and they're all sat down watching TV, and you sort of sneaked in and started feeding them sugar, and they're going to get a bit restless. They're going to start moving around. If they're blindfolded, actually, watching TV when they're blindfolded, doesn't matter. If they're blindfolded, they're going to sort of gradually sort of walk into each other, but they're going to gradually spread out. And this sort of thing happens just by complete random chance. A key word that I'm just going to drop in here, and it just means the random movement of particles. Brownian motion. Okay, and if you look that up, it just literally means the random movement of particles. So the closer those particles are together, the more frequent the collisions are going to be. Every time they collide, they're going to bounce off, they're going to move away from each other. And they're going to spread out until we have the lowest number of collisions we possibly can. And that happens with any fluid. It's going to happen with liquids. It's going to happen with gases. So if we start thinking about some of the places where this comes up, if we're looking in animals, let's go, I don't know, let's do it. If we're thinking about animals, we could do it like this in the exams. We could actually, there's no reason we can't sit there and we can't just literally do a quick mind map and try and get a head around what's going on. We can do it in the intestines. It happens on a cellular level. Cellular level. In the lungs, we've got oxygen going in. We've got carbon dioxide. In cells, we've also got oxygen. We've got carbon dioxide. Oh, let's do that over here. There we go. Can you see that on there? Yeah, just about. Intestine, we're going to have other substances. Now, I'm going to put water with a star next to it, because that's going to be mainly osmosis when we get to it, but it is related. We are going to have nutrients. So we are going to have some of our food types. Now, active transport will play a part here as well. But we are going to have sort of amino acids, we're going to have fatty acids, we're going to have glycerol, we're going to have glucose, we're going to have other substances as well. But there will be a combination of diffusion and active transport in these places. If we then look at plants and we do a sort of similar idea of where diffusion is going to happen, we're going to have in the leaf and the roots as the main ones. So the roots, again, I'm going to put water down with a star. You can argue that we're going to get some oxygen and carbon dioxide transfer. That's going to be really minimal. That's going to be mainly for sort of respiration. In the leaf, we're definitely going to have water, but it's going to be water vapor. And that's quite key because there's no star next to this one. This one's not osmosis. We're going to get some oxygen and we're going to get carbon dioxide moving in and out as well. As well as a few other substances, but again, some of this will be active transport. So, movement substances, high concentration to low concentration. Now, I don't think I actually copied the exam question out on the one I was going to show you on this one. No, I didn't. That's fine. One of the sort of simplest exam questions on this would be screen's got a bit blurry for you, is it? Is it just you, Tanishka, or is that everybody? Let me close a few things down just to make sure it's not my connection at the moment. Okay, I've shut a few things down. If it's still happening, let me know. I've got another couple of bits and pieces I can probably shut down. Okay. Okay, so I've just moved everything up for a minute. So the sort of, I suppose, the basic question on this one, and they don't bother drawing most of the sort of... Okay, brilliant. Thanks, Poppy. Thanks, Mia. They don't bother bringing in most of the cell organelles at this point. They will probably maybe just show you the nucleus, and they'll just assume that everything else is there. And what they'll probably do is they'll probably do something like this, and they'll do loads of dots around the outside. So let's just do a load of these for a minute. I'm going to get it really quick. Okay, and they'll just label these as something like oxygen. And they'll just get you to talk about which way diffusion is going to happen and why. So all you would need to say is that actually the oxygen is going to diffuse into the cell because we have our low concentration inside and we have a high concentration outside. And that would be all you'd need to say. Put it as sentences, do it as bullet points. So no problems with that one. But just keep it really simple. Oxygen moves in because there is a high concentration out of the cell and a low concentration into the cell. Right. Osmosis. So osmosis is the diffusion of water, and that's really, really key. So we've got this idea. In fact, I'm going to type this just because it'd be a bit quicker for you guys, so you don't have to wait for me to write while I'm with this. So we're looking at diffusion of water across a semi-permeable membrane. You can change semi-permeable for partially permeable if you like. There's not a problem with that one. But the really key features with this one is the fact that we're looking at it's got to be water and it's got to be across a membrane. So if it's any other substances, it's not osmosis. Those two things are absolutely critical. Now, when we're talking about this idea of the diffusion of water, if I just draw a very similar diagram to what I did a minute ago. So let's just say we've got a cell of some sort. Right, let me just make that a little bit bigger. So very similar to what I did a minute ago with this. Okay, but I'm going to label it as something a little bit different now. Let's just make this a solution of some sort. So let's just label this as, let's just call it salt. Now, we have a high concentration outside of salt. So, and this is really the bit, I suppose, that students make some really big mistakes when they're talking about osmosis. We've got a high concentration of salt. But that equals a low water concentration. Okay. Inside, we're going to have the opposite. So we're going to have a low concentration of salt, but we're going to have a high concentration of water. So in this case, what's going to happen is water, and I'll just change my colour again, water is going to move out of the cell to dilute down the solution around the outside. Okay? Just say that just again, just to make sure you've got your head around it, because it's really, really important, and it's probably the biggest mistake that students make. So if we've got lots of salt around the outside, we've got a high concentration of salt outside, that means it's going to be a low water concentration. So if we've got a low concentration of salt inside, that's a high concentration of water. So water is going to move outside of the cell to dilute down this solution that's surrounding it. What this is going to do, this is going to make the cell shrivel up. Okay, it's going to make the cell shrivel up, and we would call this flaccid. So water's moved out, the cell has swiveled up. If we have the opposite of this, okay, if we have the opposite of this, oh, press the button again, there we go. Which in this case, we're going to have our nucleus, we'll do loads inside, and only a few on the outside. Nice and quick and easy. This means we've now got a high concentration of water outside and a low concentration inside. Okay? In this case, water is going to move into the cell. So water moves in. For an animal cell, to start with, the cell will become turgid. So let's just imagine that's about as full as it can get. But in actual fact, what it's really going to do after that is burst. And we call this lysis. And it's actually going to destroy the cell. We've actually burst it. Yeah, it's just going to get destroyed. Now, it's one of those things, if your kidneys aren't working properly, one of the things that can happen is you actually your lungs start filling up a little bit with water because it's not been able to get out of it. We've got some fairly weak cells in our lungs, and those cells can burst, and we start getting other problems as well. So if the kidneys aren't working, what we would generally do is we'll be put on dialysis, where a machine does the job for the kidneys. Okay? Plant cells are a little bit different. So let's say we've got our plant cell. So if we've got, let's just say we've got a higher concentration, I'm going to say of water. Hopefully we've got our heads around, that means that's going to be a low salt concentration. This way we're going to have a lower by saying lower, that means from inside the cell. Okay? Now, in this case, if we've got a higher concentration of water, this is outside of the cell. This means the water is going to move into the cell. So in this instance, it's going to move in. In this case, it's going to move out. Now, because plants have a cell wall, they don't actually burst. They can't burst like animal cells do. So in this case, they will get as big as they possibly can. And then that's it. And they will become turgid. And if you look at plants after you've just watered them, and they've got plenty of water, and they're nice and healthy, they're looking big, they're looking upright. Yeah, there's no sort of wilting or anything else, and that's when all the cells are turgid. If we look at this idea with a lower concentration of water outside, that means the water is going to move out. So our cells are going to shrivel up. We've got a cell wall around the outside still, and they become flaccid. And actually, when we look at our plant in this condition, the plant's going to look really wilted. It's going to look really shriveled. It looks like it needs a good drink of water at this point. So plants work slightly differently. They can still become flaccid, like animal cells do, when water moves out. But they can become turgid rather than bursting, which is what animal cells do, because they've got that cell wall, and the cell wall's giving it structure, it's giving it support, it's stopping them from bursting. So really key thing with this one, when we're looking at osmosis, it's only water, and it's across a membrane. Notice up here, I sort of put down that for plants, we've got this diffusion of water in the leaf. Now, if I was to have water vapour inside the leaf, hang on, here, this is our gas. Because the water's moving around, it's going to leave through the stomata, and we'll come back to this in biology too. Because it's not going across a membrane, it is not osmosis. Okay, it's leaving through a pore, it's leaving through an opening. There is no membrane involved, so even though it's water, it is diffusion rather than osmosis. So you need to make sure that you don't make that mistake. Right, final one is active transport. And I'll type this one again as well, just because it's a little bit quicker. Oh, sorry. So, we're looking at movement of substances from low concentration to high concentration using energy from respiration. So, when we look at osmosis and we look at diffusion, just go back over. These two are both what we call a passive process. Okay, it's a bit like rolling a ball down a hill. It's going to happen one way or the other. It's not a problem. We don't need to physically do it ourselves. It's just going to work. It's just going to roll. Again, I'll put it up here as well, just so we've got it. Diffusion is very much a passive process. Those particles are going to naturally spread out. Now, the problem we've got when we look at active transport is some substances and minerals in roots, particularly key on this one. We don't have very many of them in the soil, but the plant still needs them. So the key bits I suppose we're looking at is we're going from low concentration in this case, rather from a high concentration. And we're trying to force them together. So we're going to push them together to where they've actually got even more collisions. And when we're sort of talking in terms of a topic that will come up in A-level if any of you do it, called entropy, they don't really want to go that way. I'm using the word want in a very loose. Obviously, they're not alive, but it's not a system that favours. It's a higher energy level. So they want to try and go to a lower energy levels with the least number of collisions. So in this case, what we actually have to do is exactly the same as if we want to push our ball back up the hill. We need to provide energy for this to happen. So I'm using energy quite loosely here because obviously even with the ball rolling down a hill, there's an energy transfer. We know that. I'm doing this more of a model exercise at the moment just so you can get your heads around it. So low concentration to high concentration, we're forcing them together, and this is going to take energy from respiration. There'll be particular structures that are sort of taking those minerals, they're pumping them through, and they're forcing them together. So this is not diffusion, and that's the really key thing. Diffusion and osmosis is very passive. When we're looking at active transport, we are looking at an active process, not a passive process. This uses energy. Okay? Hopefully that's quite clear on those differences. Now, a question that came up that some of my year 11s, they sort of struggled with a little bit, I suppose, is, let me just bring the question in, or part of the question in, it's this one here. What it was talking about is why, when we look at animals and plants, we need to have particular structures, particular organs in there, which, turn the volume down a bit, particular structures, particular organs, which are actually designed for transporting substances. So, if you think about your body for the moment, if we want to try and get oxygen to our cells, there's loads going on in this case. We've got muscles which are moving, they're using energy. They are making our thorax, our chest cavity, bigger. And by making it bigger, it's increasing the volume, and oxygen is being drawn into our lungs. We then have particular structures in our lungs, and we're going to look at these when we come on to, again, biology 2, called the alveoli. And they are specifically designed to have a huge surface area. And that surface area is designed to get the oxygen from the air into our blood supply. We've then got the whole of the circulatory system, which is designed to then pump that blood to smaller and smaller blood vessels, to where it can then diffuse through, again in capillaries, got a massive surface area, to where they can then permeate through into cells. So there's loads and loads going on. When we look at prokaryotes, bacteria don't need this. They're much, much smaller. And the two sort of structures I gave you here were sort of bringing in some fairly basic level maths that was designed to try and explain, actually, in terms of surface area to volume, if you can think about the fact that bacteria are really small and there's only one cell. But when we look at large multicellular organisms, they're huge. And if we're trying to actually get gases from the outside to the inside and other substances, we need to do more with it. And that's what this was proving the point with. And so to do this, what they wanted to do was to work out the surface area to volume ratio for these two cubes. So in this case, if we were sort of working out the surface area of cube 1, we're looking at each side as 1 by 1, which we know equals 1, and there's 6 sides. So our surface area is going to be 6 centimetres squared. When we're looking at our volume in this case, our volume is going to be 1 by 1 by 1, which is 1 centimetre cubed. So our surface area to volume is going to be 6 to 1. Okay, in this case. If we look at cube 2, we're looking at our surface area. We're looking at 2 by 2 for one side, and there's 6 sides. So 2 by 2 is 4. 4 sixes are 24 centimetres squared. Okay. If we look at the volume, 2 by 2 by 2. 2 times 2 is 4. We're looking at 8 centimetres squared in this case. 2 by 2 by 2. 2 by 2 is 4. Yeah, that's right. Sorry, I'm in one of those moments at the moment. So our surface area to volume ratio in this case is 24 to 8. Now, if I can just bring up so if we then go through and we actually look at the ratio in science, what I would do then is divide both these by the smallest number. We end up with a 3 to 1 ratio. So our surface area is much, much smaller for the volume. And obviously, as the organism gets bigger and bigger and bigger, these values sort of matter quite a lot. So this has a lower surface area, so we're going to struggle to get as many substances in. We're going to struggle to get the rate of diffusion as fast as it can be. No questions. Now, factors affecting the rate. So, if we want to increase the rate of diffusion, and this sort of comes up loads and loads when we look at different questions, there's not too many answers. It's about applying pretty much the same thing to different circumstances. Sorry, just have a quick. If we're looking at increasing the rate of diffusion, the first thing we want to do is increase the surface area. And any sort of structure that's designed for the fastest rate of diffusion possible will have the largest surface area possible. If we're looking in animals, we are generally going to have quite a rich blood supply or a transport mechanism for removing those substances. If you want diffusion to happen constantly, the thing we need to be able to do is to maintain the concentration gradient. Okay, so again, if you can imagine that ball rolling down a hill, the steeper the hill is, the faster it's going to happen. Now, as things will diffuse, so if I just go back over to, where is it? Yeah, let's go over to, say, this one for a minute. If we've actually got the water sort of moving, let's do this one. If we've got water moving out of the cell, this is going to keep happening until the concentration is the same. The concentration is the same inside and outside of the cell. So if we can keep removing sort of the water from the outside of the cell, this will keep happening. So if we want diffusion to happen as fast as possible, what we want to do is maintain that concentration gradient. So we often look at when is water loss the fastest in plants. So we're looking at dry and hot days. And generally with a breeze, because what that breeze is going to be doing is blowing the moisture away from around the leaves. So as the water diffuses out of the leaf, it gets blown away, which means the concentration of water outside of the leaf is always low. That maintains the concentration gradient and means diffusion is always going to be happening faster. So we're looking at maintaining the concentration gradient. In general, if we're looking at transfer of gases, it's often moist. We've often got, because it's easier, if there's a layer of water, it's damp, then gases can actually dissolve into that water much faster, which is what's making this happen a lot quicker. Okay? Right. How long have we got? About 10 minutes. So, one of the required practicals that comes up here. Okay, so we've got one of our required practicals is looking at osmosis. Now, it's a fairly basic one, and I think it's one of those ones that once you've got your head around, it's absolutely fine. The exam boards did manage to confuse students by making a few changes, and they'll do this quite a lot as they go through. Remember that 40% of the marks in any exam will be applying your knowledge to an unfamiliar situation. It's about taking what you know. It's about then working out how you can apply it and how it's being used, and using that in something that you haven't seen before. And these are the sorts of questions that after the exam, students turn around and go, you didn't teach us that. It's like, well, actually, no, we didn't. But you did learn all of these different bits and pieces. So one of the questions that came up recently was sort of an axolotl, which had the diffusion membranes for it on the outside of its body. Lots of students didn't twig to start with that all of the question was about diffusion. And if I can find it, I'll bring this question in next lesson so you can actually have a look at it and you can see. Lots of students kept talking about breathing. Well, breathing is a function to do with lungs. This just had these structures on the outside, so there were no lungs, so it didn't breathe. And lots of students were struggling to get their heads around that. But I think the ones that did well was when they noticed that actually the question was about diffusion, and all they had to do was apply that knowledge to that situation. The required practical on osmosis is the same. So the practical itself is quite simple. What you end up having is a series of boiling tubes, test tubes, vessels of your choice. So we'll just have 4 of these for the moment. And inside this, you are going to have a root veg, or a piece of root vegetable of your choice. Now, Generally, in schools, we work with potatoes because it's easier to deal with. But I think the year that lots of students got confused, they changed the carrots, and students couldn't realise it was exactly the same practical. These will all be the same size, so we'll try and cut them to the same size as much as possible. And then what we're going to do is we're going to have some salt solutions or sugar solution. It makes no difference. So let's just say we've got a 0% salt solution. This is our high water concentration. Then let's just say we've got maybe 2%, 4%, and 6%. If we're looking at our water concentrations, this is going to be a high concentration of water. And the concentration of water decreases as our salt solution increases. So what you would do is you would weigh the potatoes beforehand, or the root veg of your choice. So you would put them into the same volume of the different salt solutions. You would leave them for a set amount of time. After that, so we would leave them we would then remove them. We would pat them dry to make sure that we're not working out the mass of the water on the outside of them. And then we would re-weigh. Okay, we're going to calculate the mass again. And the really important one with this one is we're going to calculate the percentage mass change. Again, I've seen this in exams before where students miss that bit, but by working out the percentage mass change, it doesn't matter as much what the starting mass was. Now, although we're going to be trying to take them from the same vegetable if we can, we're going to be cutting them to the same size, so they've got the same outer surface area, so they've got the same volume. They will probably have small amounts of moisture difference to start with inside them, so there will be slight differences in mass. So by working out the percentage mass change, it doesn't matter that they all have slightly different masses to start with. What we're then going to do is we're going to plot them on a graph. So let's just go to here. We're going to have our y-axis. And we're going to have our x-axis somewhere like that. So on the y-axis, we are going to have percentage mass change. And on this side, we are going to have the concentration of our salt solution. And we're going to plot these. I'm not going to do this particularly accurately, but we're going to plot them maybe like that. Depends on what the solution is. And we're then going to draw our line of best fit, which is going to be something. Actually, that's going to bug me. Sorry, let me just change that. Start here. It's going to be something like that. So, key things to note about this is anything sort of, this is going to be an important area there. We'll talk about that in a minute. Anything this side means the concentration outside potato or veg cells is higher than inside. Okay, and that's the concentration, I'm going to put of water, not of salt. So make sure you've got your heads around that, and if I'm confusing you, please put in the comments, because I'm more than happy to try and clarify any of this. So do let me know. Anything this side means the concentration of water inside the cells is higher than outside. Okay, so in this case, water is going to be moving outside of the cells to try and dilute the solution down. This here, this equals the concentration inside the potato or vegetable cells. Okay, so that point here where there is zero mass change should tell us what the concentration is inside those cells. Hopefully that makes sense. I've moved a couple of times from the salt concentration to the water concentration, but hopefully I've managed to sort of clarify that so it does actually make sense to you. If it doesn't, again, let me know. I'm more than happy to come back. I'm more than happy to look at that again. But it's quite a common exam question, and those sort of points on that graph, they're what the exam board's going to be asking. They're what you need to be able to talk about. You need to understand in your head why it's happening, but it all comes down to osmosis. We are looking at the movement of the water in this case, not of any other substance, which is why the potato or the vegetable of your choice is getting heavier or it's getting lighter. Okay? Right. I'm going to leave it there for that for today. I don't want to do any more because I want you to make sure that's actually quite clear in your heads. If I just move this up for a minute, and let me just move this over. So these are the notes that we've got today. I will save this and I will put it onto the Google Classroom for years 9, 10 and 11. And hopefully that will be of use. I will drop some exam questions. I'm not going to do many because obviously I've set Seneca as well into Google Classroom as an assignment for you to have a go at. What I'm going to do on Wednesday, I'm going to cover microscopes. I'm going to cover aseptic testing, so working with microorganisms. I'm going to cover cell division, and I'm going to finish off the cells topic. And if we have time, I'll move into biology 2 as well. What I will then do, probably on Friday, just because we're going into half term, I'm going to do the second half of waves, which is most of the triple science work. So that's going to be the structure of the eye. That's going to be the structure of the ear. That's going to be ray diagrams, convex and concave lenses. Okay, so that'll be slightly more in depth, but I want to make sure I get everything prepared properly for that, because that's quite an important one for you to get your head around for those of you that are going to be doing triple. Any questions, feel free to post on Google Classroom or message me on ePraise or anything else, and I'll go through and help. Enjoy the rest of the day. I hope you're all well. I hope you're all staying sane, staying inside. But let me know if there's anything else I can do to help. Take care.